Qalcova and Jemvelva actives are two well-known and trusted active ingredient insecticides from Corteva Agriscience. They belong to the spinosins class of insecticides, a unique chemistry derived from the naturally occurring soil bacterium Saccharopolyspora spinosa. Spinosins A and D originate from the naturally occurring fermentation processes of this soil bacterium, making the first commercial spinosin insecticide of natural origin, Qalcova active. Spinosins J and L also originate from the soil bacterium and were followed by two very small chemical modifications, making possible the discovery and development of the second spinosin insecticide, a naturally derived product called Jemvelva active. The spinosins, Qalcova and Jemvelva, have a unique mode of action, different from other classes of insecticides. To explain this better, in the insect central nervous system, action potential firings travel from a sending neuron cell, pass through the synapse, and continue on through the receiving neuron. Like many insecticides, the spinosins target the insect's central nervous system. More specifically, they target the receptors of acetylcholine, which is a major excitatory neurotransmitter released into the synapse that sends firing action potential messages from the sending neuron to the receiving neuron. Normally, acetylcholine binds to nicotinic receptors on the receiving neuron, temporarily opening acetylcholine channels and allowing entry of sodium ions to complete transmission of the neuronal firing. Enzymes are then released into the synapse to degrade the acetylcholine molecules, clearing the synapse. The channels close again, ending the firing. Insecticides that target acetylcholine receptors work by disrupting this sequence. Insecticidal molecules, here represented by the yellow teardrop shapes, also bind with the acetylcholine receptor and open the sodium channels, but they are ignored by the enzymes that degrade the acetylcholine molecules. Thus, the insecticide molecules remain attached to the receptor and keep the acetylcholine channels open, allowing continuous entry of the sodium ions. The result is continuous and abnormal hyperexcitation of the insect's nervous system, ending in paralysis and death of the target insect. So how are the spinosins different from other insecticides that target acetylcholine receptors? Other classes of insecticides that target acetylcholine receptors, represented here by the yellow and blue colored shapes, activate the nicotinic receptor by binding at the same acetylcholine binding site, thus competing directly with acetylcholine for occupancy of the receptor. However, the spinosins, like Qalcova and Jemvelva, represented here by the green triangles, activate the nicotinic receptor indirectly by binding at a site that is different from the acetylcholine binding site. They do not compete with acetylcholine for occupancy of the same receptor as other insecticides do, keeping the channels open and thus allowing the continuous entry of the sodium ions and the continuous abnormal action potential firing. Based on the spinosins' mode of action, the Insecticide Resistance Action Committee assigned them to the Group 5 mode of action classification. The spinosins are the only members of that group. Because of their unique mode of action, the spinosins have shown no cross-resistance with other classes of insecticidal chemistries. These attributes make Qalcova and Jemvelva actives excellent tools in building strong insecticide resistance management programs, and great solutions in agriculture to manage damaging insect pests for efficient and responsible food production.